Hello, my name is Kamei Quarters. I work in Dr. Jackson's lab at the University of South Carolina-Aiken, and my project is cloning and testing the effects of a short interfering RNA on the HIV transactivator of transcription. Within the HIV genome, there are 3 structural proteins, GAG, POL, and ONG, 2 regulatory proteins, TAT and REV, and 4 accessory proteins, VIF, VPR, VPU, and NEF. My project focuses on TAT, which stands for the transactivator of transcription, particularly at the sequence 5834. How TAT works is once HIV has been transcribed, the viral mRNA will be translated in the cytosol where TAT will be produced. TAT will then relocate itself back into the nucleus where it will bind to new viral mRNA at the TAR hairpin. Once it is bound, it will recruit the positive transcription elongation factor B and phosphorylate the RNA polymerase. Once the polymerase has been phosphorylated, it will increase viral transcription, which will increase the viral mRNA concentration within the cell. TAT is a very crucial component for viral replication, therefore it is a potential target for antiviral therapies. The approach we would like to take is by utilizing the RNA interference pathway, which is a natural cellular pathway or process that is designed to silence mRNA at a specific sequence. This leads to our hypothesis that because TAT is such a crucial component for viral transcription, anti-TAT short hairpin RNAs should trigger the RNA interference pathway, which is expected to inhibit TAT-dependent expression of a reporter gene. The first step into utilizing the RNA interference pathway is by creating a double-stranded short hairpin RNA. Since my target is TAT, my design is TAT DSH 5834. In my design, I have my target, which is my sense, the hairpin, and the antisense, which is complementary to my sense sequence. The sequence was manually converted from RNA to DNA for transcribing purposes. We will anneal the 2 strands together for the second step of our project, which is cloning. And Figure 2a shows the plasmid we will be working with, which is PH1 stuffer minus. To use this plasmid, I must first remove the stuffer sequence, which is shown here in red, by performing a double digest with the restriction enzymes HND3 and BGO2. Once the stuffer sequence has been removed, I can then clone my shRNA into my PH1 plasmid, creating my final plasmid, PH1 TAT DSH 5834. To verify that my cloning step was successful, I will perform a PCR reaction from the M13 reverse to the M13 forward region to confirm that my DSH 5834 is within that region. After cloning, I will test the shRNA silencing efficiency by performing a beta-galactosidase reporter assay. In a 6-foot plate, 500,000 cells will be transfected in the presence and absence of a TAT-dependent beta-galactosidase and in the presence and absence of the shRNA. Once after transfection, the cells will be fixed with formalin and xGAL will be added. The Figure 3 shows the mechanism of when beta-galactosidase is present, it will cleave xGAL, leaving a blue pigmentation in the cells. Figure 1 shows the results of the annealing process. The anneal was expected to be 60 base pairs, and since it lies right underneath the 100 base pair mark, I can say that my annealing was successful. Figure 2a shows the results of the double digest that was performed on the PH1 stuffer. In lane 1, we have our DNA ladder, and in lane 2 shows the separation of the plasmid from the stuffer sequence. The top band is the plasmid, which was expected to be 4,220 base pairs, and the bottom strand is the stuffer sequence, which was expected to be 977 base pairs. Since the 2 bands have been separated and meet their expected base pair mark, I can say that I successfully removed the stuffer sequence from the plasmid. After cloning my shRNA into my PH1 plasmid, a transformation was performed. From that transformation, a colony was selected for PCR testing, which is shown in Figure 2b. Lane 1 is the DNA ladder, and lane 2 is the PCR fragment from the M13 reverse to the M13 forward. It was expected to be 483 base pairs, and since it lies right underneath the 500 base pair mark, I can say that the PCR reaction was successful. Not only did we perform a PCR reaction from that colony, but we also sent it for sequencing to verify that my shRNA was successfully cloned into my PH1 plasmid. When the results were received, we were able to identify the sense, the hairpin, and the antisense from my original design. Therefore, we can say that my shRNA was successfully cloned to make my final plasmid PH1 TAT DSH 5834. Figure 3 shows the results of the beta-galactosidase reporter assay. The positive control contains the TAT-independent beta-galactosidase. The negative TAT reporter contains the TAT-dependent beta-gal in the absence of TAT. The plus TAT reporter contains the TAT-dependent beta-gal in the presence of TAT. The plus TAT minus SH contains the TAT-dependent beta-gal in the presence of TAT, but in the absence of my shRNA. And the plus TAT plus shRNA contains my TAT-dependent beta-gal in the presence of TAT and in the presence of my designed shRNA. From each well, we were able to collect the average by counting 5 fields of view and counting the X-gal positive cells. From this, we observed that the plus TAT reporter and the plus TAT minus shRNA were statistically significantly different. We expected these two to be around the same average, or our plus TAT reporter to be the same average as our plus TAT minus shRNA, since they both had the presence of TAT. But an important observation that we took from this was that our plus TAT and minus shRNA and the plus TAT plus shRNA were statistically significantly different, which was what we expected to see. These results lead us to our implications, which state, since we observed a significant difference in the plus shRNA well compared to our minus shRNA well, our designed TAT DSH 5834 did indeed reduce TAT expression. In summary, we designed and synthesized the oligonucleotides to form the TAT DSH 5834, successfully cloned the TAT DSH 5834 into PH1, performed a transfection, and recorded the average positive X-gal cells from each well. In future work, I will clone my second shRNA, which is TAT 5860, into PH1 stuffer, and repeat the transfection comparing the two shRNAs and their efficiency to silence TAT expression.